I am going to bring up a more advanced topic now. It is important to know which end is which in DNA because they use these ends in order to replicate and in order to know how to read itself. Think about it this way. Whenever you guys read, so in our culture, the way that we read is from left to right. You know that information I give you on the first page of a textbook should be read first before you read the last page of that textbook. So left to right is the processor. Another way you can see this is up to down. You wouldn't start on the bottom of a textbook or a document before you read the top part. So DNA also has a method and how it's read. It has to do with the chemistry of DNA. So the two strands, now I'm going to teach you guys, if you can see here, one of these strands, let's look at the strand on the left side. There's that phosphate sugar backbone, repeating monomer units, and there's their nitrogenous base hanging off on the side. If I look at the complementary strand, it's upside down, if you will. And that's because it won't completely mirror the other strand, more so than it is anti-parallel to that strand. So think about it as kind of like upside down, it kind of flips. So if the phosphate happened on one side, it won't start with a phosphate over here. It actually starts with a hydroxyl group, OH, and it ends with a phosphate. Whereas over here, it ended in hydroxyl. So it's called anti-parallel. This is important. So biologists can label DNA strands. They can find the end of one, the beginning of the other. We call this prime ends. And we give the numbers 5' and 3'. When I say prime, I'm talking about that little notation seen here after the number. So 5' end, and this would be the 3' end. So the ends, the prime ends, are actually based on the sugar molecules within each strand, and the phosphate ones as well, what's attached to it. So what I'm trying to tell you guys is that if you have a phosphate that ends that DNA strand, that's the end or the beginning, however you want to see that. So the 5' end, you will always find a phosphate group at the 5' end. Appreciate that on our left strand is that phosphate group. Over here on the right, the 5' end is also a phosphate group. The 3' end is going to be the hydroxyl group that's kind of hanging off of that sugar molecule. So here's the 3' end, hydroxyl, OH, here on this strand, 3' end, hydroxyl group over here on this side. It's important to know these ends because as DNA is being replicated, new monomer units are going to be added. The strand is going to keep adding those nucleotide monomer units until it's done being replicated. They are only added at the 3' end. DNA nucleotides are only added at the 3' end. They will not be added where that phosphate group or 5' end is, only at the 3' end. That hydroxyl group is what adds new nucleotide monomers. Maybe right now, an advanced question, you can think about why that is. Why would monomers only be added at the 3' end? Hopefully that OH will help you guys. I did teach you about two different reactions. One is involved in building large molecules, and one is involved in shortening large molecules. So I'm talking about dehydration and hydrolysis, right? So why would that OH be important for this concept? So this is an advanced topic and I'm not going to test you guys on. I just want to teach you why the number 5, why the number 3. Why did scientists use these numbers? Why isn't any other number? It has to do with the chemical makeup of the sugar. Deoxyribose is the sugar in DNA. At the 3rd carbon, we use proper rules for nomenclature. Organic chemists have come up with this rule, and so this is the 3rd carbon in this sugar molecule. Well, the 3rd carbon, then, the 3' carbon, is the one that has the hydroxyl group, the OH, that I was just pointing out. The 5' carbon in sugar is going to be the one that's responsible for attaching the phosphate group. So that's where the numbers 3 and 5 come from. So kind of recapping that question, let me see if I'm recording. Thank goodness I am. Okay. So bringing back that question that I asked you guys, where we're considering why is it the hydroxyl? Why is it the OH, where only new monomers can be added? So we look here really quick. Just want you guys to focus on, get my pointer, this nucleotide up here. There's my phosphate, quite large, envisioned here. Here's a sugar. I can see that hydroxyl group, there's a 3' with that OH attached to the side, and here's my base, one of my nitrogenous bases. I can see here that a new monomer is wanting to get added. Here's another monomer, another nucleotide that wants to get added. I don't see that nucleotide being added here by the phosphate of that already existing monomer. I see that OH wanting to connect with the phosphate of the next monomer. So really what's going on is that this is the existing monomer that's going to create a new bond. Here's the hydrogen that's going to get let off in that reaction, and there's also an OH that's part of the previous phosphate group that gets let off. Well, check it. We made H2O, water. Water leaves the reaction in order to form a new bond at the 3' end of the already existing templates. This is a dehydration synthesis that I taught you guys. We're elongating the polynucleotide chain. The elongation or the lengthening will only occur on the 3' end of the already existing polymer. Notice how they attach. Dehydration, since water is leaving that reaction, I now have a connection to the new monomer. There's that new bond that's being formed. Now I have an even longer polynucleotide. This would be a dinucleotide that I'm showing you guys because I have 2 nucleotides. Appreciate that in this linear chain, you'll always have, no matter how many monomers you add, you'll always have a free prime end. There's that phosphate group hanging off to the side here. That's a free prime end. And you'll have another 3' end that's available to attach new monomers. Right, Paul, if one DNA strand has the sequence 3' TAC, ATA, CA, 5', what is the sequence of the other strand? So as you're thinking about this, I hate to talk while you guys are thinking about it, you can pause. But to kind of give you some help here, it looks like I'm going to give you the same sequence. ATGT, ATGT is the same in this response here. And that's true. If I go ahead and really quick tell you from parent, T will bond with A, and going by those rules, so it'll be ATGT, ATGT. That doesn't change. The new concept I'm giving you guys is this 3' end and this 5' end. So what is its complement strand going to read? The answer is B. Remember that the other strand is anti-parallel. So what I mean by that is it's going to get flipped a little bit. So the ATGT, ATGT doesn't change. Those nitrogenous bases are going to bond in that way. But it's kind of flipped in a way where if you had 3' on this end, the other end has to be 5'. It's anti-parallel, like it's flipped a little bit. There's an OH at this end. There must be a phosphate at this end of its complement, just like in the previous image. So it must start with 5', and then it'll read the way that we just discussed from left to right. And then it's actually going to end in 3'. You can think of it as the opposite of that 5'. It's going to end in 3'. Well, that's answer choice B.